Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we are thrilled to announce that our ecosystem is growing and are delighted to count Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and Montana Molecular as our ecosystem partners. Become an ecosystem member yourself and join our partners and your colleagues today. The ecosystem is your GPCR-focused virtual playground. Join over 400 of your peers who have already started exploring, connecting, and collaborating better. You can explore the ecosystem by signing up and getting a free site membership. For the next couple of weeks, once you sign up for your free account, you can watch the talks from the third edition of the Dr. GPCR Summit for free. When ready, you can also get a paid membership to unlock the ecosystem's full benefits. If you'd like to register your team or company, or if you live in a developing country, please reach out to us by email or on the ecosystem, and we'll be happy to help you join us. The list of benefits of the ecosystem is quite long, but today we want to highlight that you can get direct access to our ecosystem partners, ask questions, and who knows, find your next job in the ecosystem. Our job board is a GPCR-focused one where you can explore different opportunities, and if you are looking to hire, you can submit your job description. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Explore the possibilities by navigating the site using the direct links in the footer. Check it out today at ecosystem.drgpcr.com. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm delighted to have with me Dr. Stéphane Anger from the University of Toronto. Good morning, Stéphane.
Good morning. Thanks for having me.
Thank you. It's so great to have you on. Before we jumped on the call, you were mentioning that my emails ended up in your junk. I'm glad that I was able to reach out to you and have you on today.
No, I'm also glad. I'm sorry about this. My inbox is getting out of control in the past few weeks, so I'm sorry about that. But I want to say that I'm listening to almost every new episode you're doing, and I was just saying before we started recording how commendable what you're doing is, and how important it is for the field, but also for scientists that don't have the same privilege as others, right, to attend meetings and to get the training. I think this is a fantastic initiative that you're doing around the field of GPCR research and science in general. So thank you very much. Thank you very much for doing this.
Thank you so much. And it's part of the mission is really breaking down barriers, physical barriers, but also kind of leveling the field independently of who you are and where you are. As long as you're interested in GPCRs, this is a good place to tune in and also come on as a guest. I've been trying to get trainees and students and postdocs to come on the podcast, and I'm putting it out there. Everybody's welcome, and you don't have to be intimidated. I don't bite. But I'm excited to have you on this morning, Stéphane. Would you please walk us through as to who you are as a scientist and where you started and to get to where you are today?
Right. So, I grew up in Quebec City and just went through the typical education, right, of high school and then cégep in Quebec, right, which is a 2-year period that you do before high school and going to university. And I have to say, I was always interested in science, and I was always a good student. But what was driving a lot of my choices back then was I was a student athlete, and I ended up playing university hockey at McGill University, which as I said, most of the choices I was making were sports oriented and not so much studies. But when you go to university, I had to choose a program, and because I was drawn to science, I had to pick a program, and I decided to go do a biochemistry degree at McGill University. And after that, then after my undergrad, again, I had some opportunities maybe to go play professional hockey in Europe or in the US. But it was pretty clear that I was not going to be, a top player, and ended up making a career out of it. So I was I had to start thinking a bit more seriously about what I wanted to do. And I love science and I love my degree. So, I think it was the summer before the third year before I ended my program at McGill that I had an internship at a small company called Biosignal in Montreal. And they hired me after my completion of my BSc to work there for a bit, right? So I think I worked for 6 months at Biosignal. And for those of you that don't know Biosignal, I guess they were one of the early GPCR company that, back in the days were producing recombinant GPCRs in membranes, and that was their product, right? They had maybe 50 or so GPCRs that they would sell and send people that didn't have the expertise to produce GPCRs, wanted to do some ligand characterization or radioligand binding assays. So those were the early products. Of course, they evolved from there, but that was their bread and butter, if you will, product. So I worked there, and of course, I was exposed then to amazing scientists in the field. Working at Biosignal, but also, different GPCR in academia that use our product.
Yeah.
And, when I was talking to the people at Biosignal, and once they understand my interest of what I wanted to do, everybody were saying, you got to go do some graduate studies with Michel Bouvier in Montreal. Michel is outstanding scientist, and of course, for me, I don't know anything, right? Like I don't know this field. I'm just learning. And Michael Dennis at the time, who was the president of Biosignal, was a good friend of Michel. They were collaborating on different grants. And Michael had a very good word, I guess, with Michel, and eventually I ended up in Michel Bouvier's lab, right, as a graduate student. And I spent 4 years in Michel's lab during my PhD, and at the time at Biosignal, they were investigating a novel technology that they were licensing from Vanderbilt, which is the bread technology. And when I came to Michel's lab, I said, hey, there's this new technology that Biosignal is developing. I think that would be great to maybe apply that to GPCR. And, at the time in, the early 2000s, right, or end of 1990s, building on the work of Thierry Hebert that had observed higher order molecular species of GPCRs, the question of dimerization or oligomerization of GPCRs, whether they were real or they were, biochemical artifacts, right? Because of the SDS page gels.
Yeah.
So the question of, can we try to observe this phenomenon in vivo in cells, right? And the bread technology is an energy transfer technology where you tag one of the protein of interest with luciferase and the other one with GFP. And when they are in close proximity, the GFP lights up, right? So that was, it seemed to me that it was a great approach to try to demonstrate that GPCR existed in living cells and in cells. And then, the most of your listener, I guess, are familiar now with bread, but that was the very first few experiments that were conducted in Michel's lab, and we demonstrate that we had a signal, and apply the technology to other type of protein interactions, to the recruitment of arrestin, for example, to GPCRs using this assay. That's something I developed too when I was in Michel's lab. And of course, now many other graduate students and postdoc are doing amazing things. And when I see Michel's talk, I'm blown away about how far this has gone, right? They have so many biosensors, and other people in Montreal and around the world are building amazing tools to dissect signaling mechanism of GPCRs. It's just amazing how far this has gone, and it's even used, I think, in industry too, on screening platforms and various places. So I never would have imagined that this would lead to this, but I guess this was my time in Michel's lab.
It's so interesting. You mentioned biosignal and the plasmid. So originally, as you know, I did my master's and PhD with Nicolas Seveker at Saint-Justine.
Mm-hmm.
And it was a time where these BRET biosensors were, being developed in Michel's lab. And I think the first set of biosensors we started using in Nicolas's lab were looking at CXCR4 homo and heterodimer with Yann Percherancier back in the day. And I think the original luciferase plasmids, based on their name, someone at some point told me, oh, yeah, these are the biosignal plasmids. So I never had the chance to, interact with the biosignal folks. But by that time, I remember the people telling me, oh, yeah, this is, I can't remember, it doesn't come to mind right now, but they had a specific name and people were like, yeah, those are the biosignal plasmids, definitely.
Yeah, no, I think the idea for biosignal was to try to develop that as a product, as a technology that then could sell. I don't know. I don't think it went very far at that time, but that was the idea, basically. So they had kits, right, with luciferase plasmids and GFP plasmids that people could buy and then tag their favorite proteins.
Yeah, but once you have the DNA, why would you go back and buy again the plasmid? And that's kind of how it took off. I mean, you would mail it on a piece of paper, and there you go.
No, and then from there, leaving Michel's lab after my PhD, I asked myself the question what I wanted to do, and it was clear to me that I wanted to continue somewhat doing cell signaling and 7-transmembrane receptor signaling, and a class of receptors that were poorly, I guess, characterized, at least on a signaling and biochemical point of view, were the Wnt-frizzled signaling pathway. And it's, of course, now we know that they are a super important class of signaling receptors and molecules for stem cells and tissue homeostasis and developmental biology. So I went to one of the founder of the field for my postdoc, Randy Moon in Seattle, that discovered the frizzle receptor and was one of the first person to demonstrate the importance of Wnt in vertebrates, in frogs embryo. So I went to do my postdoc there and spent 4 years with Randy, and then I was recruited back to Canada at the University of Toronto and the Faculty of Pharmacy in 2006, where I started my own lab and spent 15 years there to build my own group and continue research in the Wnt field and more broadly in cancer biology and functional genomics. And just recently, last year in September, I got the call to be the new director of the Donnelly Center in Toronto. Was a multidisciplinary research institute working at the intersection of functional genomics, stem cell biology, and engineering. So that was an experience. A vision of Brenda Andrews that many of you would know, that was the initial director for the first 15 years, that basically brought investigators from different disciplines into the same roof, under the same roof in the same building, basically to interact and use each other expertise and leverage each other expertise to further everybody's science, right? So it's a place with very strong scientists from different fields, and it's a place that is focused entirely on research. So for me, it was, if I had to choose a dream job, it was definitely something that was compelling and interesting to me. So I took up the challenge, and I've been there for a year now.
That's great. Yes, and the news, and we put the news in the Dr. GPCR newsletter. I don't know if you've subscribed, but that was something that I always hunt for whenever I see promotions and people switching jobs, especially with this kind of level. I'd like to take you back a little bit in the past. You mentioned that you were always hockey and science. for the two main interests. I think you're our first guest with a hockey history. Yesterday, I
Mm-hmm.
recorded an episode with Oliver Hartley, who wanted to be a musician, who actually took a break to see if he could become a rock star. So, rock star and hockey player. As a child, you mentioned you were a pretty good student. Did you have a preference for science, even as I'm thinking teenager years?
Yeah, I was always drawn to science topics. I remember a teacher in high school, a biology teacher that was doing extracurricular work during
Mhm.
lunchtime, and I remember dissecting. I think it was a beaver
Wow.
during lunchtime, and that was outstanding to me. That was great that we had opportunities to do that, and some other experiments looking at cells under a microscope and things like that. In high school, I think now it's more mainstream, but back in the days, doing these type of experiments were not very common, right? So, and then in Cégep, I had a chemistry, organic chemistry teacher that this guy had an amazing ability to communicate his excitement and to teach. And from that point, I was pretty sure that I wanted to do a degree in that field, right?
Yeah.
I never thought that I would do graduate studies and research long term, right?
Mhm.
I think some people know right away, but for me, it was too early. But I knew that science was important and I wanted to continue in that field as opposed to more maybe physical science or arts or something like that, right? So, that's why I decided to do biochemistry at McGill,
And
right?
Miguel.
Which to me was biochemistry was the more complete training that I could get, as opposed to maybe biology or microbiology at the time, or so that's why I decided to do that.
And you picked McGill because of the hockey team? I would
Yeah,
have said, sorry, Frank.
that was. I mean, it's a strong university
It is.
that played into the decision, but mostly, I was recruited by 3 or 4 universities to go play hockey,
Mhm.
some in the States and here, but, the combination of the program and the hockey environment was the best. You know, for those of you that follow hockey, actually one of my teammates at McGill there was Guy Boucher, who's a recognized figure now, I think, in the hockey world, with a coach in the National Hockey League for a couple of years, and he's on TV, I think, in Montreal now on RDS, commenting the Canadians, right? So he
Wow.
was a teammate of mine back then. So I met amazing people through that journey, through sports, but um, you know, also the people at McGill. Of course, it was a solid training environment. Many of my classmates are actually professor now everywhere
Wow.
in the world, right? And actually, one of them is in the Donnelly Center. Gary Bader, we were co-students together, and Alex Palazzo was another student in my class at McGill. He's a prof in the Department of Biochemistry at the University of Toronto. So we were classmates together.
Small worlds. A very, very
Mm.
small world. That's fantastic. And then how was the transition from Montreal, from Michelle's lab into Seattle? I mean, changing countries, being in an Anglophone environment, although you went to McGill.
Mhm.
Um, how did you adapt? I'm thinking here about the postdocs and the trainees who are thinking about going abroad to continue their studies. Any take-home messages from there?
The take-home message for me is do it, right? Sometimes this is nerve-wracking, intimidating, but this is probably one of the best experiences in my life. To you know, the postdoc is amazing because you don't have to worry about uh, classes and thesis and um, other obligations that you have as a graduate student. I think you're focused 100% into research. I think that was um, of course, depending on the lab, right? But Randy's lab was extremely well funded by the Howard Hughes Institute. We had, a group of about 20 postdocs, right? So it's an amazing intellectual environment and research environment that, was very stimulating to me. Of course, people find themselves at this time in their life, right? 25, 26 years old when they finish their PhD, where, they sometimes they have partners that have jobs, and um, sometimes they have family, right? And maybe kids. So there's a lot of important decision that goes into this. But I was fortunate that my wife was a physical therapist, and it was very easy for her to find another job in the U.S. So there was not a lot of hesitation, right, on her part when I said, "Hey, I'd like to maybe think about going to do a postdoc in the U.S." She was immediately on board with the idea. So that component for me was easy, right? For other people, it's not as easy, right? They have family ties, or the partner has a very important job career wise, and
Yeah.
other components comes into play. But the idea of being faced with something new is always good, right? Challenge yourself to not being complacent. um, Push the boundaries of what you can do. I think those are really important components that people can do or should do once in a while. And coming into a relatively new field, right? My knowledge in developmental biology and stem cell biology were not very good. Um, was humbling to me, and to face or to be in a group of 30, or so very strong postdocs. You realize that, wow,
You have
there's a
some
lot
work
of
to do.
good people out there. Yeah,
Yeah.
exactly. So, but it was great. And Seattle, of course, that's an interesting question, right? Because I talk about that with my own students now, where they're thinking about postdocs, and they ask, or they think, where should I go, right?
Yeah.
Should I go to the Bay Area, or should I go to New York, right? I tell them that it shouldn't be the way you approach it, right? So it's not where you want to live. It should be, what type of science do you want to do? Who are the investigators that you know are doing the type of science that you see yourself doing? And then the second aspect should be the city, right?
Yeah.
But that being said, right, I think the city you're in also plays, depending on your life condition, whether you have kids, whether you have a partner, and if there's other um, components that goes into this decision. I think it's an important component, but at the end of the day, you need to find a subject or a field of research or a technology or something that that's the main reason I want to do this, right?
Yeah, and there's also the component of chemistry with your future boss and your lab mates. So I think going there and visiting and talking to people and reaching out to people who went through the lab is also a great way to investigate if this is a good fit for you.
Yeah, no, of course, the track record of the individual, what have the postdocs that have gone there before you, where they end up, right?
Yeah.
The type of interaction that the person provides, the type of environment that the funding environment, the ideas, the project, right? But I think everybody is looking for something different, right?
Yeah.
In that aspect, right? For me, I was looking for a place that had the resources, right? That but the of course the mentorship that I needed was more on the writing piece, and
Mhm.
the all to I mean, Randy is an amazing writer, and the ability to tell a story in terms of his papers, and that's the part that I took from Randy, was his ability First of all, he had one gift, or he realized really early, I don't know, one of the two, that the currency in science is papers. And
Yeah.
he was never the rate limiting step into returning manuscript to students and postdocs. So, you would submit a manuscript to him, and basically the next day, you would get the revision on your desk, right? And he was asking, okay, when do I get it back? And that's somebody that had like 30 people in his lab, right? And that's somebody that was at the top of his field at the time. But he was never the rate limiting step. I mean, I cannot do that myself.
It's
Right now
hard.
I'm trying to, but for him, he identified that as a priority, and he was really, really, really fast and efficient at returning manuscript. Anyway, I took more than that from the
Yeah.
time in his lab, but if I can think of something that was quite unique to him, I think was his ability to write and to return manuscript and to get the work into the pipeline.
Which I think is also interesting, but you made a great point about what you took away from your time in your postdoc and what you identified as a need, which was your need to learn how to write manuscripts and how to tell a story. But I think that also requires some self-knowledge and acknowledging what are your areas on which you have to work. And I think that's very important
Yeah,
too.
I mean, it's this is all in hindsight, right? It's not
Of
like
course.
when I left Michel's lab, I said, "Oh, I cannot write. I want to go
Yeah, yeah.
to a postdoc lab." That it's in hindsight what I think I got, right? I mean, Michel, for people that know Michel Bouvier, he's an amazing storyteller as well, right?
Yes.
But he also had his own style of writing manuscript, and I took a lot from him. He taught me everything, if you will, right? My fundamentals came from Michel, and then I went to Randy's lab and I saw Randy's style, which was very different from Michel. And then what you do is you pick up what works for you from your different
Exactly.
mentors, and then you frame who you are, right?
Yes.
And I think that's basically what I was trying to say, right? So I think you know um, the influence of mentors into
Exactly.
shaping the type of scientist that you are now is what I guess I was trying to say.
No, it came through. It came through clearly. I was just trying to point out, especially to the trainees listening, is that sometimes it's worth sitting down and making a list of what are your strengths, what are your weaknesses, and really think about whether you want to collaborate with someone to, help with those weaknesses, or go get some training in order to gain those skills.
Yeah.
I have another question.
What
Yes, please.
Yeah, sorry. I just wanted to. The other thing also for me, when you asked the question about how did I choose the postdoc lab, right? So, in Michel's lab, I had a very solid training in molecular pharmacology,
Yeah.
signaling, cells, right, culture, and things like that. So, and biochemical approaches. But, it was pretty clear to me that I wanted to learn model organism, and Randy, what is famous for his use of xenopus embryos and zebrafish embryos and genetic techniques to study developmental biology aspects and signaling aspects. And because they were studying this, poorly defined class of 7-transmembrane receptors, I thought that was the ideal fit, right? That address in terms of training what I wanted to do. So that was the main reason why I decided to join his lab.
Which was actually my question, is why developmental biology, but then the connection was 7-TMRs in model organisms and developmental biology.
Yeah.
When was the first time where you realized, oh, I'm working on GPCRs and they're so cool to work with? Was that the time during your time at Biosignal, or once you ended up in Michel's lab?
Yeah, I think it would be when I was at Biosignal. So at the time, I was an intern or a summer student or an employee when I finished my BSc, and oftentimes you're pair with a group leader in a
Yeah.
company, and I had the amazing opportunity to be paired with Roger Bossé. Many of you would know Roger. I think he's pretty famous now in the industry for his development of the alpha screen, and um, he's just a very robust scientist, right? So he had come from Marc Caron's lab actually as a postdoc, and he was just arriving, right, fresh from his postdoc at the top of his game, full of excitement, right, full of energy, and here I am, right, a pretty naive young scientist with this guy that was able to communicate his passion and to teach me, right, the basis. And I remember one of my first tasks as an employee was to characterize the new product that Biosignal wanted to sell, the PACAP receptor at the time,
Yes.
which was a particularly difficult one because of the hydrophobicity of the ligands and things like that.
Yeah.
But working with Roger was amazing, and he became a close friend. So, and it made me realize, of course, the importance of the protein, the GPCRs, and all this, and their roles, but also just the what you can do as a research scientist, right? So
Yeah.
I remember he was working on the final revision of a neuron paper that he had done in Mark's lab, right? And he was telling me about it, and I just thought it was fantastic. And I'm glad that our paths crossed. He was
He
a very important influence on my career.
he ignited the need
Exactly.
to answer questions related to GPCRs. What's your favorite GPCR?
That's a good question. I mean, I have to see the frizzled receptors now. This is what I've done for the last 15 years or more, 20 years of my life, is to work on this class of receptors, which we know a bit better how they work.
Yeah.
Of course, you mentioned GPCRs. The G protein aspect of frizzled receptor and signaling is it's getting clearer, right? But it's a
Yes.
controversial topic, I think. But it's pretty clear now. There's very solid genetic evidence now that suggests that G proteins are not involved in signaling downstream of Wnt and frizzled, at least right for the signaling pathway downstream that we understand the most, which is the signaling through beta-catenin, the beta-catenin pathway, right? So there's been CRISPR-based knockouts of GPCR of G proteins that basically suggest that they are not involved. So that's pretty clear. That being said, there are so-called beta-catenin independent pathways that are downstream of Wnt activation of frizzle that appears to require G proteins, and they may be a component. The problem there is that we still don't have accurate and robust readouts of signaling to study these beta-catenin independent pathways. But and the evidence that suggests that they are important in some type of physiological roles, there is some scattered evidence that are pretty strong, I think. So I think there is a role for G proteins downstream of frizzled, but there's not been a lot of work in this area, mainly because of the lack of assays and the lack of readouts that are robust enough, I guess. Yeah.
Of tools, yeah. The question is, what is your favorite GPCR? But it could be a G protein or a receptor family, or to push it to the extreme, like Andrew Tobin did. He had a phosphorylation site as his favorite phosphorylation site, which I thought was very interesting. So you mentioned, Wnt and frizzled. So there is 10, almost 11 frizzled receptors, 19 Wnts. It reminds me a little bit, and this is a super naive question. It reminds me a little bit of, the chemokine receptor system. Yes, they're class A receptors, and you have 20 receptors, 50 chemokines. With the tools that we have today, do we know which Wnt interact with which frizzled, and what are the signaling or functional consequences?
No, that's basically the short question. and it's not because of the lack of interest, right? It's again because of the lack of tools. It's getting better, but we don't have purified Wnt proteins for all 10, right? So maybe there's two. Those are very hydrophobic glycoproteins that, for the most part, stick to the membrane, right? And so it's been very difficult to do typical pharmacology type of experiment and to try to dissect, which Wnt binds to which frizzled. And in fact, there's a lot of pleiotropic interaction, right? So one Wnt will bind to 4 or 5 different frizzled. There's a couple papers out there, if you look, right, that have tried to address this question in a systematic way. Mm-hmm. But it's mostly using overexpress type of approaches. Yep. So you have to take it, I think, with a grain of salt. I think now with the advance of CRISPR type of technologies, the requirement for different frizzled in various processes is the way to go about things. And at endogenous levels of frizzled and endogenous levels of Wnt being secreted and having the right temporal and spatial right expression of these receptors, I think, is really what dictates the ultimate response.
What so, you mentioned a need for new tools or for better tools to really dissect this interactions. If you had a magic wand and you could create any tool to help better understand Wnt frizzled function and interactions, what would that look like?
Well, I think it's coming, right? And my lab has been working hard on this front to develop new tools and new reagents, basically, to study this pathway and to potentially manipulate this pathway for therapeutic purposes, right? So, we've been involved over the last 10 years or so in developing antibodies, recombinant antibodies against every frizzled receptors and every LRP5 and 6 receptors. So, we had amazingly productive collaboration with Dev Sidhu, who's a recombinant antibody expert, right? So a phage display expert. In Toronto, we shared a grant, a Genome Canada grant, which the purpose was to identify antibodies against all cell surface targets for cancer biology, right? And Dev identify experts in different field, and my role into this was to do the frizzled LRP5/6 pathway. So over the years, we accumulated amazing antibodies against all 10 frizzled LRP5, LRP6 that really start to provide the set of reagent that can detect where they are expressed. You can block their function. You can study the individual role of the frizzled receptors in various model organism in mice and stem cells and so on, right? So I think those were important missing tools, and now we can do this. Maybe 5 years ago, we then repurposed these antibodies into agonist, basically. So the way that the frizzled and LRP system works is through heterodimerization of frizzled and LRP, right? So the role of Wnt ligand is basically they act as a chemical crosslinker, if you will, right? So they bring the frizzled 7 transmembrane receptor and the single pass LRP5 and 6 together. And this is sufficient for activation, right? Wow. So, there was just an important paper by Bill Weiss published in Science Signaling this week, and I wrote news and views for the paper where, unlike GPCR, where the allosteric communication between the ligand and the G proteins is very important for the signaling mechanism of GPCRs, there doesn't appear to be allostery, right? At least for the beta-catenin pathway, at least for the binding of the most proximal effector disheveled to frizzled, there's no allostery that is needed, right? So, in this case, it's mostly like cytokine receptors, where just the clustering of frizzled and LRP, which is promoted by Wnt, is sufficient to initiate signaling activity downstream. So, you know, that started. I remember a postdoc project in the lab where we said, okay, well, we're good with Dev at developing antibodies. Maybe you can generate a bivalent antibody where one arm of the antibody would bind to frizzled and the other arm would bind to LRP. And, the idea there would be that you would induce the clustering of the two receptors, and this would be an agonist, right? So the postdoc, Yu Yong, worked for 2 years to make this work, and nothing worked. They were always behaving as very potent antagonists. Mm-hmm. Presumably because the distance, right, of the two arms of the antibody was not high enough to capture simultaneously the two receptors. So they were always finding one or the other and block the pathway. So Yu Yong, who was a protein engineer himself, tried all sorts of new combination and decided one day that if the distance is the problem, maybe I can put the two paratopes on each side of the FC, right? So rather than putting the two binding sites on the same side, like a conventional antibody, he decided to put the frizzled on the normal side, but then he grafted on the other side of the FC, the LRP 5 and 6 antibody. And then it worked, right? I remember the day that he brought me the results in the lab. I couldn't believe it, right? Because I knew the implication of that discovery. Michel used to say, it brought tears to my eyes, but I think that result really did, right? When I saw that, I was amazed, and I was really happy about the possibilities that discovery brought. So essentially now, because we had already a set of antibodies for the 10 frizzle receptors, when you graft these antibodies into the agonist format, now we have selective agonists for all 10 frizzle, right? We can activate all 10 frizzle at will, very selectively, in any system we want, right? So that basically, I think, is transforming the ability to study the pathway. And of course, I recount that story, but as it's often the case in science, you think you're brilliant, but at the same time, there's a lot of smart people out there, and it turns out that there's a lot of people that were thinking about these things, and the pioneer in the field, right? Royal News and Cleavers, right? And had already kind of figured out a similar type of antibody system, which is quite different from the ones we do have. But these type of surrogate Wnt ligands now are making their way right into the field and are transforming the ability to study this pathway, which I think this is a welcome addition. The other thing, of course, that really changed everything in the last few years is CRISPR. The ability to knock out at will, genes of interest, signaling molecules of interest, to study their function and their requirement, right? The requirement test, I think, is really the gold standard, right? To define the role of a protein or the role of a gene. And also the ability of CRISPR, of using CRISPR and gene editing, basically to tag endogenously a protein or a gene of interest, right? Yeah. So you can put GFP, you can put a flag tag or bioID type of reagents at the endogenous level. And it turns out that this is key, right? Because once you start overexpressing things, especially in a pathway like the Wnt pathway, that everything works through protein-protein interaction, then you start creating things that normally do not necessarily occur. But with the ability to tag endogenous proteins, then you solve that somewhat, right? And this is changing the way that we can study this pathway and everything else, really, right? Like not just wind and fizzles, but this has changed the whole biomedical sciences.
Yeah, being able to see what happens at an endogenous level in a model organism, I think it's necessary. And now having these tools is just a blessing when you think about it, because previously, and this always came up whenever you were working on any protein, but on GPCRs as well, you would transfect your cells, and then the reviewers would come back and say, "Oh, yes, it's an overexpressed system." But being able
Yeah,
to look
I mean, I
in,
wouldn't dismiss anything, right? I mean, I think there was important discoveries that
of
were made
course,
because of overexpression. Yeah, like
of course,
inverse agonism and the ability to do binding and things like that. But
yeah,
at the end of the day, right? It's I think the sufficiency test
yeah.
is what matters,
Yeah.
right?
I think it's two
Yeah.
sides of the same coin. I think it's complementary information.
Yeah.
You need your hex cells, but you also need your model organism. You also need to look at your receptor function in a more native like environment.
Yeah, I meant to say the requirement test, right? Is your gene of interest required for a
Yes,
specific readout or phenotype or signaling
exactly.
output? Yeah.
Exactly. I also have another question, and I think the answer is typically yes. I'm pretty sure your answer is going to be yes as well. Do you think GPCRs are still good drug targets? But I would like you to expand on that a little bit.
Yeah, no, I think we're just scratching the surface of the potential of this family of proteins, right? I mean, I think one area that has been completely understudied is their role in cancer, for example, right? And there's a lot, there's more work being done, but still, I don't think enough. Their role in the immune system,
Yeah.
and modulating the immune environment for cancer therapy and others, right? I think this is another area that I think is really important. You would know, right, with your background on chemokine receptors.
Yeah.
Um, I think there's 7 transmembrane receptors that we barely know what they do, right? The whole family of adhesion receptors. There's more getting known now, but then I'm following all of this field with a bit more of a distant eye now. But it seems that there's been breakthroughs in that field,
Yes.
right? And there's others, right? There's a number of GPCRs involved in metabolic diseases that are coming up. So I would say that it would be pretty simple-minded to think that this is over and that GPCR is not an important drug target, right? I think, of course, there's a tremendous amount that has been done, but I think there's new opportunities that are always there, and they are proven drug targets, right? So
Exactly.
I think the ability to target this family of protein, they're exceptionally druggable, right? And now I think we see a lot more of antibodies type of approaches to target GPCRs, right? Which I think it's took a long time for whatever reason to use antibody engineering to target GPCRs, but you see a lot more of the use of antibodies and nanobodies and variants of antibodies, right, to
Yeah.
target GPCR ADC conjugates, right, and things like that.
Yes.
So I think we're going to see a lot more in that front. Yeah.
Agreed. I think the field and the biotech or the private sector is definitely leaning towards developing therapeutic antibodies using phage display, machine learning, in order to target GPCRs using antibodies. And one of the big questions is when you pick a receptor that you want to work on, is what do you need? Do you need an agonist, an antagonist as an antibody, and what disease context also?
Yeah,
Okay.
no, I think it's very complex, of course, right?
Yeah.
The decision that goes on into the pharmaceutical industry and the decisions and the IP space and the diseases and the market opportunity. And I've been part of company creation and been part of different companies over the last few years. And that made me realize a few things about it's not always the best ideas, or there's a lot that goes on into these decisions.
Yes, yes, and then there needs to be not only traction, but there's this culture of fail fast so that we can move on to the
Mm-hmm.
next thing. But
Mm-hmm.
yeah, there are a lot of questions, so I think we're not going to be out of work when it comes to drug discovery or working on GPCR biology for sure. We've talked about it a little bit about advice for junior scientists who want to contribute in the field. We've talked about, thinking about what you want to do and how to pick your lab. Anything else you'd like to add to that?
Um, you know, I you I often tell students, right, that you need to figure out a way to distinguish yourself. There's a lot of people, I think, that do that, right? That do graduate studies and postdocs, and you got to figure out a way to people remember you somewhat, right? And it could be through your involvement into various initiatives, or it could be through outstanding science, or it could be through your participation at meetings, right?
Or starting a
I,
podcast.
or start a podcast, right? Like do something that people are going to say, oh, that's the person that did that, or this is this person. I remember that person at this meeting, right? Or
Yeah.
I ask questions, right? Like I'm always amazed when at meetings that people are intimidated or afraid to raise their hand and ask questions. And I know it's a barrier to get to that, but you know I remember as a graduate student, right? I mean, I grew up in the Bouvier lab with Ali Salapour, who
Yeah.
you may know. Ali is a professor now at U of T. He was in Mark Caron's lab during his postdoc. Amazing scientist and friend, right? But as a graduate student, I remember going to an industry GPCR meeting in Orlando with him, and we would literally ambush all the experts in the field, right? We would go and like ask them questions and, hey, what do you think about this? What do you think about that? And talking to Mark Caron and Graham Milligan and Tuwe Schwartz and you know people that were my heroes back then,
Yeah.
right? And it was amazing, and really, these people remembered us, right?
Yeah.
Because we ambushed them in a way, right? And when it was time to ask for postdocs, and they knew literally who we were, and they knew our work, of course. But I think so what I'm trying to say, I guess, is don't just go, by the motions, right? Figure out a way how to distinguish yourself from the pack, right? From the herd, and
Yeah.
do something special that people are going to remember, and carve your way, carve your own way, right? And you're a good example of that, right? I think
Thank
with
you.
what you've done, not only with your science, but this initiative that you started. I think it's a one example of how you can do something that is transformative. Yeah.
And interestingly, that was the first thought behind it. I wanted to continue working in the field or enable the field without working in the lab. And that was
Mm-hmm.
very important for me. And the podcast idea and interviewing people opened up so many doors. I've met so many phenomenal scientists, people whose papers I read, papers I've cited, and now I interviewed them and we formed these relationships. And I think this is amazing. And it's kind of a multiple benefit type of endeavor where I get to meet people like you, but also I get to, you know, disseminate this information and share these conversations and potentially inspire other scientists to
Mm-hmm.
contact you and ask for a postdoc or have these discussions. And it's important. And I like the way you put it about distinguishing yourself from the herd, and it's important. And yes, it
Yeah.
takes courage, but you can definitely do it and raise your hands.
Mm-hmm.
And yeah, I know Ali is wonderful. I'm trying to get
Mm-hmm.
him on the podcast, so now it's recorded, and hopefully he's gonna come. He's been hesitant. He said, I don't work on GPCRs anymore." I said, "But yes, you do have a connection to the field, so you absolutely have to come."
The other the other thing, um, the other recommendation is also don't be afraid to put in the hard work, right?
Yes,
Because you know people often say you need to be lucky, right? But I don't really believe in luck, right? Like you have to create your own luck in a way. And you know the more you do and the harder you work, and you have to be serendipitous, I think, right? You need to be able to read a lot of paper and to recognize a good result and where this is going to take you. But you know it's just like anything else, right? Like if you go fishing and you have three lines in the water, your chance of catching a fish is going to be higher, right? So
exactly.
and I think you have to develop a passion in anything you do. And when you're passionate, then it's not work. Right? Like for me, I never felt like I was working long hours, right? It was just what I wanted to do.
Yeah,
And then it's easy, right? And
exactly.
I think, and it's not unlike other fields, right? This is not just unique to science. I think it's in any type of environment and workplace that if you want to do something significant and grow to the company, you need to be passionate about what you do and you need to put the hard work, right? So
Agreed. Agreed. I
yeah.
know we're almost running out of time. We've talked about that result that brought tears to your eyes. Any other aha moments in your career?
Oh, that's the one with the agonist with the physical antibody is definitely one that so I can think of one at every stage of my career, right? So that would be one in my own group, right? That I can think of. There was maybe a couple other ones, but this is definitely one. Of course, during my graduate studies in Michel's lab, the breath assay, I think, was the first few times that I remember running into Michel's office and, "Hey, Michel, I think it works," right? And Michel almost jumped on his desk right at that time. He said, "Oh my God, this is fantastic!" But this was one, right, and the possibility to study protein interaction in living cells in
Yeah.
a high throughput format and the arrestin assay, all of this was definitely a milestone of my graduate studies. During my postdoc, that was this is amazing. Like when I talk about serendipity, and where to, I guess, follow your gut instinct, right? So, I went to Randy Moon's lab to study the wind pathway, and to study how to use model organism and loss of function in xenopus and zebrafish. But that took a long time, right? And I was 2 years into my postdoc, and I didn't have anything. Like
Yeah.
I was at the time I had two kids, and you start thinking, what am I going to do? I don't know if it's going to work for me. There's companies that were starting to ask whether I would be interested to go, and you start thinking about this. But I came back essentially to what I was really good at, and it was biochemistry,
Yeah.
it was at the time of the beginning of mass spectrometry to do IP and mass spec, right? And I was fortunate to have a good friend in Seattle at the time, Anne-Claude Gingras, that had pioneered a lot of the tap tag methods and
Yeah.
protein complex isolation and mass spec. So I talked to her and I said, "Hey, maybe I can apply these type of technologies to study the wind pathway." And I did that, and it worked super nice. And we were some of the first people to do IP mass spec in mammalian cells and identify novel components. And there was another another investigators on the same floor as we were, that his name is Ning Zhang. He's a world acclaimed structural biologist that had solved a lot of the structure of E3 ubiquitin ligases. And basically, he saw what we were doing. And at one point, he stopped me in the hallway and he said, "Hey, listen, I have this amazing structure of this Col4 E3 ubiquitin ligase, but we're missing the family of adapters, if you will. E3 ligase, they all work with, they have the core E3 ligase, but then they have a family of adapters that then recruit substrate, right? So for the Col1 family, it's the F-box proteins, right? I don't want to go into too much details, but anyway, the Col4, they were missing this family of substrate-specific receptors for this E3 ligase that he was studying. So he said, can you use your methods and mass spec to try to identify this family of adapters? I have a hunch that they exist, right? So we did, a few experiments, and within a couple of months, we identify a family of about 60 proteins, novel genes that were not characterized, that ended up being the decaf family, that is this family of substrate-specific receptors for this family. That turned out to be really important discovery that I was published in Nature just towards the end of my postdoc. And to this date, that's probably the easiest paper that I published, right? So it took about 6 months of work. No rebuttal from the reviewers. The paper went straight into Nature.
Wow,
right?
That's phenomenal.
Of course, we had an amazing structure from Ning, but the discovery of this novel class of E3 ubiquitin ligase that today, led to the, you're probably familiar with the PROTACs, right?
Yes.
And one of the target is cereblon, basically, that is used in the PROTACs family of small molecules. So cereblon was identified as part of this paper as the substrate-specific receptors for the Col4 family. So anyway, those are three defining, I think, discoveries over my career that I can think back and say, wow, this was pretty cool.
That's awesome. That's awesome. Yeah. All right. Thank you so much, Stefan, for your time. I really enjoyed our chat.
Likewise, it's been fantastic. Thanks for the time, and thanks again for what you're doing.
Of course. Thanks so much. Thank you for joining us and listening to this Dr. GPCR podcast episode. I'd like to thank our guest, our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and Montana Molecular. Thank you to our Dr. GPCR team as well, Attila Forrest, Ines Pinero, and our newest Dr. GPCR proteges, Montserrat Avila Zozoya and Nipuna Wirasinga. Welcome to the team, Monse and Nipuna. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any question and suggestions at hello@drgpcr.com. Until next time, stay safe, and we'll see you in the ecosystem.